let's begin. So it's a really great honor to have the privilege of introducing Michael Emerman. I don't feel very legitimate to do it. There are many former labs of many persons from the lab of Michael here. Maybe Emermanitos and Emermanitas, can you make some noise? So you see, well, it's a lot of pressure, but well, it was difficult to pick one of you, so I had to do it. Sorry, Michael. So Michael received his PhD in cellular and molecular biology in '86 from the University of Wisconsin-Madison. From '86 to '89, he was a postdoc at the Institut Pasteur in France. He used to work on the biology of HIV-1 and HIV-2. Well, he still works on the biology of HIV-1 and HIV-2. And I heard he often defines himself as a person who trained in the labs of 2 Nobel laureates. Maybe he will tell you more. Then he moved to the Fred Hutchinson Cancer Center in Seattle, where he started his own lab. And over the years, he became a full professor. So he has a very long CV. I'm not going to tell everything. But, well, first he received numerous awards. So I have selected some of them. The McDougall Award for Mentoring in 2012. The Tijan Retrovirology Prize in 2017. NIH Award and others. He was elected fellow in the American Academy of Microbiology in 2016. Michael's commitment to the scientific community is simply amazing. He has supervised 27 graduate students, 16 postdoctoral and medical fellows, and a number of them have opened their own lab. He also supervised more than 80 undergraduates. He has participated in 105 thesis committees, can you imagine? And what is really incredible, so I don't know you, but each time I met somebody who has worked in the lab of Michael, he was, well, in heaven, ecstatic. I don't know how you would say. Well, so he also served as editor-in-chief or editor of numerous journals, reviewed about, can you guess, 800 articles since '89. He worked in several NIH sections and grant reviews committees. He has organized meetings, of course, Keystone, Cold Spring Harbor. He was a member of many scientific boards. And, well, he has accepted to give no more than 143 seminars, invited seminars. And, well, he has, of course, taken a number of responsibilities at the local and international level. And, of course, last but not least, there is science. And science has been exceptional. In the course of his long career in retrovirology, Michael has published at least 166 papers. But, well, what is very exceptional in my opinion is that every single paper, even in the smallest journal, is super interesting. And I can tell you because I read not the 166, sorry, but I read some of them. And yeah, well, so his discoveries are diverse and important. I will just mention a few emblematic examples. The ability of HIV to infect non-cycling cells other than macrophages, the central role of the capsid for HIV entry into the nucleus, the demonstration of the cytostatic activity of VPR, the development with Amit Malik of the concept of indirect paleovirology, and all the ensuing studies on several restriction factors, the deciphering of the molecular events at the origin of HIV, and more recently, the development of original HIV CRISPR screens for the identification of cellular factors involved in viral latency. So, well, I will leave it to Michael to tell us more about all this, much better than I can. Thank you, and thank you very much, Michael.
Oui, merci Florence pour cette introduction. So I'm going to tell this story in 3 parts. I'm going to tell the things I learned as a trainee and how that is going to shape my scientific career. Then I'll tell you about the evolution of the lab, things we've studied, and then I want to do the last part of talking about things that are important to me in science. And I have the thingy. So my first scientific endeavor was at the Ohio State University, where I was an undergrad. This is Ned Berman when I went back in 2018. He actually looks exactly the same as he did then. And I published a paper about osmium-8 reagents. And what I learned from Ned Berman was positivity. He was positive about everything. Every result was interesting. Everything was a good idea. Some of my studies disproved a hypothesis of another graduate student who had graduated, and that was great. That was interesting. He was very super positive about it. I also learned as a graduate that mistakes will be made, and this will be a common theme throughout my career. Mistakes will be made. We will learn from those mistakes. We will make other mistakes, and we will try not to make that same mistake, but there will be mistakes. Ned actually won a mentoring prize not that many years after I left, which doesn't surprise me. So then I went to the University of Wisconsin-Madison for my PhD. That is me with the mustache. I was in the lab of Howard Temin. I could give this entire talk about Howard Temin. So Howard Temin is the smartest, most unique individual that I will ever know and have known. David Baltimore talked about how he came to the problem of reverse transcriptase from the school of Arthur Kornberg of enzymology. Howard Temin came from it from the genetics phage school at Caltech. So for Howard Temin, there was genetics first, and biochemistry was to clean up the details. I loved teaching his paper in 1960, where he looked through the microscope, and there were foci with different shapes, and he could take virus from a foci of one shape and give it to other cells and make it take that shape. And from looking at shapes and counting the shapes and seeing how sometimes the shapes change to other shapes, he derived the provirus hypothesis. Paper was published in 1960. He was 26. He was a graduate student. He was the only author on it. He was so smart. This is the only picture I have with him. He wasn't the guy you took your picture with. This is with the governor of Wisconsin who came to look at some cells. So the things that I learned from Howard Temin. In terms of science, it was follow phenotypes. It was, you started with an interesting phenotype. And I'll get to this through other places in my talk. Like a cell, these cells have a different shape. Or these cells do a different thing than you might be expecting them to do. Or this virus does something you weren't expecting. But it's a phenotype. And you chase the phenotype. And you follow the phenotype. His way of doing science was design simple experiments and to answer one question at a time. So if I proposed something that was bigger, he said one question at a time, and then you answer the next question. And data leads to hypothesis, which leads to data. So going in to talk to him, you showed him data, and then you were off to the races, because he was asking about the hypothesis. What was the next thing you were going to do? What was that going to lead to? But every time it was data looks at hypothesis. When Howard Temin reviewed a paper, he did not actually read the paper. He just looked at the figures. And he made up his mind what the data meant. And then sometimes he would check if they said the same thing he thought. I also learned somewhat of my mentoring technique from Howard Temin. So one of which was you own your project, that he might have told you the project, but once he told it to you, it was yours. And when he asked you what you were going to do next, it meant, what are you thinking about doing next? It wasn't, he's going to tell you what to do next, because he did not tell you what to do next. He just stared at you and waited for you to tell him what you were going to do next. We had weekly individual meetings. His were on Fridays. I hated Fridays. Mine were on Mondays. And what I call it hands-on, hands-off. So you met every week. And it was his way of keeping you on track, of knowing, keeping you, telling him what you were doing, what you were thinking, but hands-off in that he didn't tell you what to do. He told you what he thought about it, but he didn't tell you what to do. Good morning took me a long time to figure out what that was about. Every day he walked in and he said good morning to each person. Every person got their own good morning. Even if two people were standing next to each other, they got their own good morning. So he is acknowledging everyone in the lab. It was your time actually to say something to him. If you missed your good morning, you missed your chance, but you talked to him. And high expectations. So he did not talk down to anybody. The first year grad students, he talked at the same level. You try to remember what he said, and then you go ask the senior grad students what that meant. But there was high expectations for every single person. I came to Cold Spring Harbor. It was my very first talk, and so that's why I'm so excited, so honored to give this talk now. I couldn't imagine doing that at my first talk. I was a second-year grad student. And I talked about this paper where we found deletions in retrovirus vectors. And this was the start of a large series of papers that would continue later of using single-round vectors to look at genetic events in retroviruses. So what was the frequency of mutations, deletions? In this paper, I found some deletions, and I also found there was a single point mutant. And I told him about the point mutant as kind of like, yeah, there's a point mutant. And he said, that's the most exciting thing you've ever found. And then we had to talk about why that was. So my first talk at Cold Spring Harbor, I met some scary-looking people. I didn't put names because I thought everybody would know who they were, but apparently they don't. But who knows who this scary guy is? Yeah, that's John Coffin. And this one? No, it's Beatrice Hahn. Anyway, I'm sure I was too afraid to talk to them at the meeting, but I've talked to them a lot since then. I've collaborated a lot with Beatrice. I sent her the photo and asked her. She said it's her. So my PhD was called Gene Suppression in Integrated Retroviruses. So my first paper, I found deletions. Then I devised a new system so I could look at silencing of promoters in retrovirus vectors. So we published a bunch of papers. Genes with promoters and retrovirus vectors can be independently suppressed by epigenetic mechanisms. I did some quantitative stuff. I compared it with different vectors and different LTRs. And this whole phenomena of silencing of retroviral vectors, it was epigenetic, there was LTR specificity, it was transcriptional, and it correlated with chromatin accessibility. So this is going to come up later because I'm going to come back to, we never quite figured this out, and they stopped working on it. I didn't work on it for another 30 plus 5 years. Anyway, that's what we work on now. This is me at my PhD defense. Then went from Madison to Paris. So at the time I was in Temin's lab. That was in the 80s when AIDS was discovered to be a retrovirus. And I became the HIV expert in the lab. I read all the HIV papers. Temin would get lots of HIV papers to review because he was kind of neutral of everything, and he gave them to me. So I reviewed, he checked on it. There weren't a whole, in the lab we worked on retroviruses as tools, that's what I did. We worked as retroviruses as models. And I was interested in retroviruses as pathogens. So my feeling was I have an expertise in retroviruses. We have a pandemic. I can do something that can help. So there weren't a lot of places that you could go to work with actual live HIV, and I didn't want to go to any of those. I had French in high school, so I thought this can't be too hard. It was. So anyway, I went to the Pasteur Institute in Paris. There was a lot of interesting people there. Does anyone know who this person is that I have to put the name? Yeah, it's Lady Di. Guy is Luc Montagnier. I guess that was a joke. Anyway, this was Luc de Montagnier. This is when I left, actually. Anybody know who this one is? Yeah, this is Olivier Schwartz. He was aggressive. What? Yeah, he is here. So I had two really good mentors. And so we published a lot. Oh, I'll get to that later. Anyway, this was when they had first sequenced HIV-2, so that's what I started working on. And I knew how to do transfections and molecular biology. They mostly just knew how to sequence. So I got to do all the fun stuff, actually. So I started working on the regulation of HIV-2 and mostly how that was different from HIV-1. I kind of switched and started working on regulation of HIV-1. So this is a paper published about the same time as Mike Malam's. His came out a little bit earlier about what the Rev protein did. So we had nice in situ hybridization, so without Rev, the envelope RNA was stuck in the nucleus. With Rev, it was out in the cytoplasm. I also did a lot of work with envelope and found the CD4 binding site by doing mutations in conserved areas. So you'll notice Montagnier sometimes he's on these papers, sometimes he's not on these papers. He is never, I guess this one he's last. But anyway, I had two good mentors. I actually didn't talk to Montagnier very often. So I'm phrasing this a little bit different. Actually, what I learned in Paris, which was very important. So what I learned in Paris was to collaborate. That we collaborated with a lot of people with complementary expertise, kind of all over Pasteur, some in Boston as well. This was not a thing that Temin's lab did. Temin's lab was mostly single-author papers just within Temin's lab. I learned the value of collaborations, which is going to be important for the rest of my career. I learned to get advice widely. So if I didn't have Howard Temin, I had everybody else. And it took me, I could learn that actually at the Pasteur, there are a lot of super smart people, and they were different places, and they would be happy to talk to me. And that people would be glad to give you advice. So talking widely and getting ideas was important. To use a different angle from everybody else. So what we did mostly in Paris was we did experiments that were kind of orthogonal to what everybody else was doing. So everybody else was doing much more directed translational things, and we could do something a little bit different, and we would try to do something different throughout the career. Give stuff away. People would write to Montagnier and ask him for clones, and he would give me the letter, and I would send out the clones. And then people stopped writing to him and just wrote to me. And I sent out hundreds and hundreds of things. And if you have anything that says ROD on it, HIV-2, it came from me originally. I sent it to somebody who might have given it to you. But any like TAT. Anyway, I just started giving stuff away. And as a result, if I asked for something, people gave it to me. And work on important things. So this was an important problem. It's kind of hard to emphasize the bleakness of those days, that people were dying all the time, and there was nothing we could do with them. People would call the Pasteur, and they would give me the phone because I spoke English better. And it was just sad. And people would ask me, like, is there a new thing? And like, there wasn't anything. It was a bleak time. So you got this sense of like, we are doing something important, which is a thing that carries forward to this day with all of us. Also at that time, Most people working on HIV could fit in one room, so it was more than 86 when I went. But this was a Banbury conference at Cold Spring Harbor in '88. This guy right here. But this was the majority of like everybody who worked on HIV biology. I went to the Fred Hutch in 1989, partly because of the mountains. I like mountains. I grew up where it was flat, and I really like mountains. This actually isn't the first building, but this is why my lab is right here on the shores of Lake Union. Mostly because they were building an HIV lab, which most places didn't have at the time, but they had got a grant to build one, and they didn't have anybody who knew how to work with HIV, so there was like 2 of us on the job market, and I got it. The other partly was that it was a very collaborative place, like people talked to each other. It reminded me a lot of McCardle. Like there were joint meetings, there were a lot of collaborations between labs. Didn't start out great. I came right from Paris. They ordered me a desk, but the desk was too big to fit in the door. So the lab was empty. I had a chair, though, that someone gave me from another lab, and I had a phone and started ordering stuff. I fired my first undergrad helper. I fired my first technician. Mistakes will be made. My first paper was rejected from Journal of Virology, and I never sent it back again. My first RO1 got a bad score. Stuff started to pick up. We made a cell line that turned blue when it got infected, which we call the Magi cells. And from these nice blue nuclei, because it's a nuclear localized beta-gal, so you could count them really easy in the microscope. We did a lot of interesting things with this. We did the particle to infectivity ratio with it. We looked at isolates which ones could and couldn't infect this cell line, which, as we now know, was a X4 virus. This wasn't actually the first reported cell line to be made, but it was the first one you could get, gave it away to 500 people. It was also the first one that wasn't contaminated with amphitrophic virus, but that's a aside, because I knew what I was doing. But anyway, I gave it to over 500 people. And it's been really, there's other ones that are better now, but it was the one that people used. And what it did for you was then rather than waiting 2 weeks to get virus enough you could do a reverse transcriptase assay in a growing culture, you could assay virus after a day or 2 days. The reason we made this is that we needed a tool. We needed to be able to assay virus quickly. And that's what this did. It gave you titers. It didn't just give you physical amounts, it gave you actual titers. So this is the lab at that time. You might recognize Vineet right here. That was our first paper. So the thing that we wanted to do with this came from this phenotype. So this phenotype was, these are microglial cells in brain sections, and what's stained here is HIV. So there's tons of HIV in these brain sections, and they are terminally differentiated. And retroviruses were not supposed to get into cells that didn't go through mitosis. That was actually part of the provirus, the validation of the provirus hypothesis was that you needed a mitosis, but these guys were getting infected. It could have been they got infected before and then differentiated, but then Mike Malin did an experiment where he infected macrophages in tissue culture, and they could get infected, which is why I thought this actually is, they can get infected after they go through mitosis. So that was the phenotype that we were following. And we validated that it actually was not anything about macrophages. It was actually a cell phenomena. You could take anything and stop them from dividing, and HIV didn't care. So this might have been the second paper after Paul Lewis joined, HIV infection of cells arrested in the cell cycle, and this one where we compared MLV and HIV directly in the same cells where one required mitosis, the other did not require mitosis. So then we started a series of what HIV protein allowed it to access the nucleus. So you recognize Ram here. I don't know if anybody can recognize Nicholas Manel. He's super young in that picture. This is me. And this is Masa. So Masa is the one who figured the whole thing out. So this is all due to Masa. What allows HIV to get into the nucleus? And he first did swaps between HIV and MLV, and he showed that capsid was the dominant marker in these. Capsid is the determinant of retroviral infectivity in non-dividing cells. He did a paper showing that everything else that we had said before was wrong, that wasn't any of those other things that we said it was. And then we had a couple other papers showing that directly that it was capsid, because we could make mutations that would change it from one phenotype to the other, and that things which bound to capsid could affect this phenomena. And this is actually one of my very favorite experiments. So in this experiment, we have an HIV that has GFP and NEF. We infect cells, and then we sort for the GFP positive cells, and then we ask where are they in the cell cycle. So you're only looking at where in the cell cycle are the cells that are expressing GFP. So with HIV, they can be anywhere in the cell cycle. So here's G1, S, and G2, meaning that those cells could get in any time in the cell cycle. Whereas in capsid, in an HIV with some capsid mutations here, the cells were synchronized. The cells which were GFP positive were synchronized, meaning that they could only get infected after a mitosis. So now this is 12 hours after infection, so the mitosis was previously, and they're all in S phase. They all synchronously go through the cell cycle until those cells have now divided by 24 hours, meaning that this actually, they required a particular part of the cell cycle to get in, and it was capsid that was determining this. We had some data which we did not understand, but now which we do understand, that it correlated with the retention of capsid of the incoming pre-integration complex, and the effect was post-nuclear, which we said in the paper, but we didn't understand at all. So in parallel, we are doing experiments with the accessory genes. So one of the other reasons we made the MAGU cells is we had mutations in each of the accessory genes, and we were asking what they did. And this is the work of Mark Rogel, and this is a very typical Mark Rogel picture, where in this case we infected T cells, and the cells with wild-type VPR stopped dividing. The ones without VPR just kept on dividing fine. If we kept the cultures long enough for the wild-type cells, eventually cells would start to grow, and those cells always had a mutation in VPR. And this is how we show that it prevents, it is cytostatic, and also in this paper we showed it causes a G2 arrest. And many other people, including people in this room, found the same thing about the same time. We studied a lot more VPR papers. VPR is a cool, very cool thing. But the study on the accessory genes led to collaboration with Harmeet Malik, so an evolutionary approach to host cell interactions. This came about from a talk I give at faculty meeting where I talked about VIF and APOBEC, and this is after Mike's lab cloned APOBEC 3G. And Harmeet said we should look at genetic conflict between APOBEC and VIF, and that's what his first postdoc, Sarah Sawyer, did. She was the first one to be co-mentored. And it led to this idea of paleovirology. That is, what we see today was caused by infections in the past, and we call this the paleovirology approach. We can look backwards by using phylogenetic trees and asking how things have changed along those trees. Since then, we've co-mentored a lot of students. We've co-mentored 12 graduate students, co-mentored 6 postdocs, We have 33 co-authored papers. This is like 4 of them. So we could identify, we could use positive selection alone to figure out the species-specific, where in rhesus TRIM5 we gave it species specificity, where we could use this positive selection technique, what was evolving rapidly, to say where it was without doing any other thing. We did some papers on tethering and VPU-NEF and how that changed because of changes in the host antiviral genes. There's a paper from Mali about how ApoBac3H lost its activity at least twice, and how the ability of primate lentiviruses to degrade SAMH21 preceded the birth of VPX. It originally arose in VPR. Anyway, that's only 4 of them. So Harmeet is a great collaborator because we don't agree about anything. Every result, we interpret it differently, and we say what we're going to do differently. But we do agree that we are eventually going to agree, and we listen to each other. And sometimes we change sides, and I start arguing what he thought, and he starts arguing what I thought, and we same agree. There's some people who are co-mentored, and they know the challenge of that sometimes. So this led to this idea we can trace, you can use evolution to trace the ancient and modern origins of HAV1. So here we have the SIVs in the old world monkeys. We have a recombination between at least 2 SIVs to make SIV-CPZ to go from old world monkeys to hominids. And then we have the ultimate transfer from SIV-CPZ to humans, and then another one from chimps to gorillas, and then to humans. So I'm not going to talk about this one. I do want to highlight this one. So this is the work of Alex Compton, where we use positive selection and the differential antagonism of apobecs that we could ancestrally reconstruct to show that the positive selection in one particular part of ApoBac3G, amino acids 128 and 130, was driven by primate lentiviral VIFs. And we could use that information to date when the SIVs arose, and it had to be at least 5 million years ago. And Welk Johnson had kind of similar results with TRIM5, saying it's at least that old using genetic conflict as well. We also looked at this jump. So between old world monkeys and hominids, a lot happened to create this recombinant SIV-CPZ. This is the work of Lucy Etienne, Gene Loss and Adaptation to Hominids Underlie the Ancient Origin of HIV-1. So here we have this region, the central region of SIV-RCM, which is one of the precursors, and we have SIV-CPZ. And you'll notice there's the VPX here. And what Lucy could figure out is that this went to this. There was not a recombination event, which is what we originally thought. There was actually just a loss and a gain. So SIV-CPZ and subsequently HIV-1 lost VPX. It can't do the things the VPX does, so antagonism of SAMH21, antagonism of the Hutch complex, because there is no VPX in between here. On the other hand, what this also did is it overprinted VIF. So now VIF has a new C terminus, and it also has other mutations up in here. And these are important because they allow the RCM VIF to antagonize the chimpanzee ApoBac3G. So that is one of the ways that the accessory protein adapted to the new restriction factor in the new host. We actually had some indication of what that was from some genetic experiments from Alex Compton, that it was going to be in the loop 5 of of VIF. But we started a collaboration with John Gross where we could really tell this, because he did the crystal structure of RCM VIF, compared it to the crystal structure of HIV-1 VIF, and showed me what was different, and what was different was loop 5. And just seeing that was like, okay, we got it. And it ended up being a single mutation loop 5 from Y84 to Q. HIV-1s have either a Q or an H, which are equivalent, but RCM has a Y. And this is what allows RCM to adapt to hominid ApoBac3G, both chimpanzee and subsequently humans. So we had identified genetically the interactions between ApoBac3G and VIF, and so this is where I'm getting back to my Temin moment of genetically we knew what was going on, but we needed the biochemistry to actually validate and get the details. And the validation and the details came from this paper from John's lab. Yen-Li Li led it on his side, Carolyn Langley and Nicholas Cesarino on our side, which showed that the amino acids that we thought were important are what we call these arms race, which is right here between ApoBac3G in blue and VIF in green. There are other members of the complex that you needed to get it here. And in a blow-up, so these are the actual contacts. We had used positive selection and the analysis I told you to identify D128, D130, and on the VIF side, Q83. So this was the validation of the genetics by the biochemistry. So not to say that we actually had got it all right. There are additional contacts that are really interesting that we maybe should have known about, but we didn't. So R15, R19. And the biggest surprise is that there's RNA in there. They're actually the only place where ApoBac3G and VIF can escape is at this hinge here, because there's a huge region bound by at least 4 nucleotides of RNA in between those two, and the genetics wouldn't have got us there. Molly O'Hanley, so this is taking a step back. Molly O'Hanley rejoined my lab after she was a graduate student, did some other stuff, came back to HIV. I'm sorry, Molly. Anyway, she came back and she decided that we needed a more high throughput way to figure out HIV interactions. So rather than just taking things that had been published and then doing genetics on them, could we do a high throughput screens to figure out everything that interacts with HIV? So I'm not going to go through the HIV CRISPR. It took Molly several years to get this to work, but it does work great. And Michael Young talked about it earlier. Essentially, uses the virus itself as a barcode for each of the CRISPR knockouts. And we have used this Molly first used this to identify interferon factors mediating interferon inhibition of HIV using 2 different strains. We subsequently have published this paper where we identified the host dependency factors for HIV, and I think this paper was published during the pandemic, maybe. Anyway, it didn't get as much, people didn't look at it as much as it is, because there's a huge table there where we did this screen with 5 different strains, and we have a list of like 150, and if you just ask me, I can look it up. Is it on the list or is it not on the list? So a lot of times, like when Henry Levine talked, his was on the list. I was looking it up. Anyway, there are really interesting tables in this. So back to my graduate work. So I actually, even though I didn't quite know it, I worked on proviral silencing in graduate school. And I had wanted to work on HIV cure for a while, but didn't know quite how to do it, or have this idea of like you need a tool, you need to do something different than what everybody else is doing. So we decided to adapt HIV CRISPR to do this. And so we are doing HIV CRISPR on latency, where we are trying to identify druggable targets to improve latency reversal agents, or improve the shock part of the shock and kill. And we've done this with a couple targets. So one by Emily Shea, who developed the system and also found a pathway called ING3 that synergizes with the non-canonical NF-kappa B pathway. And another one from Carly Gray, where she found that the integrator complex is actually the transcriptional block to elongation for both in the presence and the absence of latency reversal agents. That there is a complex that binds to the HIV promoter, both in vivo and in ex vivo, that prevents full-length transcription. And I think I am running out of time, so I'm not going to talk about what that means. So let me get to what's important. Mentoring is the most important thing I do. So this was actually after I won this McDougall Mentoring Award. You get a little bobblehead of yourself. I won another one later on. But to be a good mentor, you have to align your priorities so that what you are doing is helping your trainees achieve their goals. So you make it so that your expectation is that you are going to help them. Each side needs to have expectations. So my expectation is you are going to think, you're going to be motivated, you're going to be interested in the problem. Your expectation is I am going to help you in any way I can to achieve what you want to do. I've had 26 PhD students. This is all of them. Nearly all of them are still in science. So the ones circled in red are still in science careers. Gina went to law school, but I don't hold that against her. You need to build a team, and that team needs to understand that they need to give feedback to each other. They need to support each other. We do a lot of team feedbacky kind of things. We always go on a hike every summer, and new people have never hiked always think that it's some kind of torture thing, but then they end up like backpacking the enchantments. If you're from Seattle, you know what that means. This is the thing called the stair photo, which we take after every PhD defense, where we sit on the stairs and we take pictures. Collaborations are key. So I've mentioned a few of them, but all of my career has been collaborative. Collaborations allow you to do things that each of those labs couldn't have done on their own. So being collaborative means that you have complementary expertise, that you trust each other, that you listen to each other. And I've collaborated, so I mentioned Harmeet, but I've collaborated. I have 6 papers with Beatrice, 6 with Mario, 5 with Mike Malum, 5 with Julie Overbaugh, 5 with John Gross, 4 with Linda Chellico, 3 with Patrick Ed Brown, 2 with Patrick Mitchell, and 2 with Tom Hope. Julie Overbaugh has kind of been my longest collaborator. We share the HIV lab. This is a good collaboration because she thinks of things from the clinical side, from the top down. I think of things from the molecular side, from the bottom up. So we give each other ideas that we wouldn't each other had. I've also collaborated with a lot of other people. I may have missed some people. These are people I at least have 1 paper on, but the list is probably longer. Almost all of my papers have some other labs on them. That's the way I like to do science. The other thing that's super important is embracing community. This is the part 1. So that is, we are a community. We make an advance as a community. We need to stick together as a community. There is service to the field, which I count as constructive reviewing. That is reviewing that actually, if there's a thing that's wrong, you give them advice on how to fix it, or you tell them how to make the paper better. If you've been in the HIV field more than a couple years, I have reviewed your paper. I'm almost positive. You would probably know it. Being a journal editor, organizing meetings, organizing local works in progress, like getting people to talk to each other, to exchange ideas, is how we move forward. Doing teaching, so getting people in the next generation interested in what we do. And as I mentioned, being generous with reagents, with ideas. So ideas are free. You just give them away. And you give away ideas, you get ideas back. You hold back ideas, you don't get any ideas. All the collaboration, all the informal collaboration is about giving away stuff, both ideas and reagents. What I mean by this, part 2, is that breakthroughs are the end result of this community. There are obviously breakthrough papers. They're seminal papers, but those seminal papers, they all relied on stuff that came before them to make that. So just a couple examples of many. So lentiviral vectors being used in vivo, ex vivo, make CAR T cells to do genome editing. They came from understanding HIV genomes and its regulation. Integrase inhibitors came from discovery of integrase, all the enzymology of integrase that I never paid attention to, the role of complexes, the assay development. Lenacapavir for PrEP has this long history. So I mentioned a little bit of what we did, the roles of capsid, but there's a lot of other stuff that other people did to figure out the roles of capsid in early steps of the virus life cycle. Of course, the structures, mutations, the binding partners, all that led to this breakthrough. That this breakthrough wasn't possible without the community, and it is the retrovirus community. It is our achievements to do that. So finally, the other thing important to me is work-life balance. This is my wife, Rosemary, when we got married. This is us 32 years later. She looks the same anyway. These are my kids, Alex and Iris. So weekends are for family. Weekends we do hiking, we do biking. We do, when they were little, I was coach of the chess team. We went to chess tournaments. I was coach of the soccer team. Chess team's a lot better than the soccer team. And I want to thank, none of this is possible without the people in my lab. We had a reunion in August 2019, and almost everybody actually came back for their reunion. And these are the people who did everything I could ever talk about. And of course, funding, I have had funding from NID almost since I started. So thank you very much.
Questions.
Bonley, you had a question.
That was the shortest silence you've ever had not been able to sit through, Michael.
So just the background to that is the technique is that you ask a question and then you just stare and you just wait for someone to say something. So I didn't do that this time.
That was the first. Yeah, my question is, what do you think the important role of VPR is?
Where's Oliver? Oliver, you should. Oliver, right there, will tell you about VPR and its roles. Okay, we'll talk about it.
So seriously now, please.
Hello. Okay. I was just wondering, in that your favorite experiment that you showed a time course of 12 to 24 hours post-infection. Who designed that experiment?
Masa designed that experiment, but I believe we got the idea from a paper from Steve Goff.
It's such an inconvenient time course.
Okay. If Masa is on, Masa, are you on Zoom? He said he was going to be on Zoom. Can you unmute?
Hi, I'm Luciana. You seem like a great mentor. So here.
Oh.
So congratulations. Did you regret firing your first technician?
So this was one of the things I made a mistake about. So I thought I wanted somebody who was just didn't really have a lot of ambition, was just gonna like work in the lab and get stuff done. And I interviewed another person who had come in and had actually read all the papers that I wrote as a postdoc and was like so ahead of the game, but he wanted to go to grad school in a year. So I didn't take him. I took this other person who had no motivation, just didn't care, made mistakes all the time. The guy I didn't hire Actually did work as a technician for a couple of years in another lab, got his PhD, is now a faculty member. As a technician, he published like first author papers, and mistakes were made. So no, I didn't regret it, but I learned something from it. So now, the first thing I look for is motivation. Actually, are you interested in the problem that we're doing? And do you really want to know the answers? And that's the number one thing I'm looking for. I can teach you everything else. Yes?
Just to follow up on this, I sometimes find it, I see curiosity by students is the main driver often, like for, to get them into science and to make them really work on their projects. Sometimes, some people have it, others don't. How do you think one can best raise curiosity in people about their projects?
So again, I don't think I select for that. Yeah, that's a hard thing to teach. But that is one of the things that those are the people who I want in my lab. Yes, I select. I don't. Yes, they're selected. They are selected to be a phenotype.
Hi, Michael. That was great. Do you remember what I brought for my exam, for my general exam?
Did you bring me a gift? Was it
Not
chocolate?
gift, something you really like.
Okay, was it chocolate? That's what I usually recommend.
Do you remember gummy bear?
I don't remember.
I learned from my fellow classmates. They said, you just bring gummy bear.
Oh,
Michael,
the gummy bears, yeah.
Keep chewing, and he will never ask you questions.
Okay, that's true.
Hi, Michael.
Hi.
Michael. Oh, sorry.
Okay. Great talk. I'm a student of your student, Dr. Crompton. So my question is, what is the secret that all of your trainee, your student becomes faculty and their own lab? So what was the secret? Is it like you are very good in finding person and building a great team, or is it really the training because of training they become
So I would like to think that it is the training, so that they came in motivated. But I think that's why I went through what my training was and how I developed my mentoring is that things might go slower if I don't tell you what to do, but eventually you're going to learn if I don't tell you and you tell me and I tell you what I think about it. So it's all this, my trainees are training to think, and I think that's what does it. So they own their projects. I don't own it. I tell it to them, and then it's theirs, and I want them to do it.
So, Mike, you mentioned that great work. You mentioned that you learned very early that you need to set expectation high and the same for everybody. Does it require the same mentoring style, or it is different for different people?
No, again, mistakes were made. Initially, I thought, no, I got to teach everybody. Everybody's got to be treated exactly the same, no difference. And then, there were mistakes. You got to adapt it to each person. There are people who need more positivity than even the positivity that I usually do. There are people who need a little bit. Yeah, it's different for each person. You got to figure out what it is. So actually, what I do now is I actually ask them, what do you need? That helps.
Hi, Michael.
Yeah,
Oh, sorry. Go ahead. Okay. Thank you. Yeah, so following up on that, what you're saying, mistakes were made. First, thank you so much for bringing that up and bringing the list of seemingly failures in the beginning, because as a starting PI, it can be very hard to compare yourself with, people that are 5, 10 years ahead of you. So if mistakes were made, how were they fixed? Was it just iterations, or did you learn from someone else? How did you finally flick the switch to get the RO1 score better than it did before?
No, so I do have a lot of self-reflection, and a lot of it is myself writing down what went wrong, how I can do it better. I organized the very worst course ever organized at our graduate school. 10 years later, people were still talking about how bad that course was. And after reading the student comments, like, oh, here's what is bad. Here's, like one of the comments, I would ask a question and then immediately I would like answer it. Because I couldn't wait for anybody to answer. And okay, I got to stop doing that. And I learned, okay, I ask the question and then I just wait and wait and don't blink. And eventually someone is going to start talking. And like just how the thing was, that's just one example, but that's how I learned how to teach a good course, by teaching a really bad course. And kind of every other mistake, I can probably tell you the same kind of thing. Like how can I change this?
Hi, Mike.
Yeah.
Thank you so much. I have a simple question. Who is your favorite trainee?
They told you to say that. They're all my favorites.
Who should I pick up for the introduction? The same.
Hi, I've got a question. What was that paper you never resubmitted to JViral?
So we started mutating the splice sites for, was it Rev or TAT? We started mutating some splice sites. No, actually, we started mutating a lot of splice sites. And every time we mutated a splice site, we got more splice sites. And then we mutated those splice sites, and we got more splice sites. And the paper was a mess. And I couldn't actually figure out like what actually was the message of this paper. And I'm pretty sure that John rejected it. But anyway, so again, mistakes were made. Like we weren't making any progress on that. And that person switched to a different project and actually got some really nice papers.
Hey, Michael.
Yeah.
That was a fantastic talk. Yeah, right back here.
I can hear you, but I can't see you.
Behind John.
Hi, Judd.
Story,
I see you.
Of course. I think any good question is worth asking twice. So I wanted to ask what you thought the future of science looked like in this country, and what we can do to advocate for it.
Thank you for asking that, Judd. It was a dark time in the 80s. So the Reagan years were a very dark time. There was no money for HIV. People were afraid of everything. Like starting my lab, people would not go up to my floor. This is in the first thing was AZD, and AZD stopped working after a while. Like, I think my point is we've been through hard times before. Advocacy led to us getting over that, to getting funding up, to eventually leading to discoveries. If I could fault David Baltimore, is that he actually had a different example. So he talked about a strike between the time he wrote the paper and submitted the paper against the Vietnam War. So that was another dark time in our period where general advocacy eventually led us out of that. So I think the point I'm making is that we are not stuck and we are not powerless, and we can take steps to get us through that.